Good evening, everybody. How's everyone doing? Good, great, good. So it's a real pleasure to welcome tonight our speaker, Manu Prakash. Manu did his PhD in applied physics at MIT, and then after that he was a junior fellow at the Harvard Society of Fellows. He subsequently was awarded 2016 a MacArthur Fellowship. Manu's lab is really interested in basic curiosity-driven research, understanding how computation is done in soft matter systems and in living biological systems. His enthusiasm for science is infectious. I think if you can see, if you went to the demonstration table out there, I saw scientific discoveries actually being made from soil scooped out of the Gurley courtyard. But along the way for doing this curiosity-driven research, Manu's group has developed tools that enable the global community to do science, to democratize science. This not only requires the ingenuity to develop these tools, but it also requires the dedication to distribute them worldwide. So it is a way for the global community to do science, and also some of the things have global health applications. So I think not only the ingenuity and the dedication is required, but the empathy for the human condition is evident in Manu's work. On the Stanford Medicine website that announced his MacArthur Prize, it has a banner which says, "We are all part of the equation." And I think Manu is a big part of this equation, as are you for your support of KITP and science in general. So Manu, I'll pass it to you, and thanks for coming.
Thank you, Mark. Everybody can hear me at the back? It's an absolute pleasure and a delight. I can't remember when I was last at KITP, so it's just so much fun. I am going to share some things about wonder today. Just the capacity to wonder, thinking about these sets of questions in a biological perspective, but also what does it mean for us as a society to be curious? What does it mean for everybody to experience science directly? I'll do that through some amount of work that we've been doing, but at that same time, I think what's valuable to take away some lessons is how important it is sometimes for us to actually share that capacity to wonder. I titled this Recreational Biology, and you'll see in a second what I mean. And Mark might not know this, but most of the other people might know this. I am not known to finish my talks on time, so I start with acknowledgements. I don't work alone. It's a remarkable group of young individuals, students, postdocs, and just phenomenal collaborators around the world. They're spread around the world, and it's just really a joy to do work as a community and as a collective. I'll talk about individuals that have been involved in specific discoveries, but really this is really an incredible collective effort. So let's start with the title. You know, I think tongue-in-cheek that it may be, recreational mathematics has been a very powerful tool for us to discover new vistas in mathematics. I was so delighted. I saw a t-shirt somebody was wearing, "Without geometry, life would be pointless." And like, that's the gist of my entire talk. And I want to make a link today between recreational mathematics and how we find problems in recreational mathematics, why they appeal to people, and could we apply those ideas in biology as a whole. So the history of recreational mathematics is phenomenal. It goes all the way back to 2200 BC. I'm assuming, how many of you have read something written by Martin Gardner? So I think if you haven't, this would be your homework assignment. You could start with his article on Conway's Game of Life. Just a remarkable writer who was not actually a trained mathematician, but had the capacity to see the power of puzzles and use those puzzles to explain very deep ideas in mathematics. And sometimes when, at least how I was taught biology, I was just thrown at a lot of information and I couldn't make sense of it. And I think it's time for us to revisit those sets of ideas and see, could we actually learn something from mathematics as done and apply it to biology? So I think, this is really the gist of the talk. Niels Bohr, a letter that Max Delbruck wrote in his end phase of his life, actually hints on this idea. He said, I will run into paradoxical situations analogous to what classical physics runs into in the attempt of analyzing the atomic phenomena. This, of course, has been my ulterior motive in biology from the beginning. And he was really talking about searching for paradoxes. And I don't know how many of you know that Max Delbruck is a remarkable biophysicist in his end of his life. And he was working on fungus. And he was working on how fungus can detect light. So phototaxis in fungus. It still remains to be an open problem. And he would just do these remarkable experiments, very much focused on a paradox that he had discovered along the way. So how do we take that idea and apply that to a broader sense? And I think I've already mentioned, I often like to start with my favorite books. This is one of my favorite books. You can watch why, and all of us, just when you look at nature, can be enamored by it, and this squint in the eye, sometimes we find that same squint in the eye when we stumble upon sets of phenomena. And actually, that is really at the heart of much of what you end up doing in science. Now, ironically, when you ask this question and you look at life as a whole, I know there are some astrophysicists probably in this room. I'm not trying to compete with the incredible variety of stars that you have, but I do want to point out that 99% of all of life taxa still remains to be discovered. So for the mere earthlings that are stuck here, and some of you who have chosen to pursue a dream in biology, there is still a lot to be discovered. And I'm not talking about species. I mean, this is literally taxas. Like the entire trees of life might actually be completely missing because we just haven't had the capacity. Most of this is microbial in context. And one of the things that we've been thinking about is microbes, every little species is literally an idea. So the first mathematical link that I want to make is, what is my number line? The tree of life is actually a number line. If you were to really think about a described tree of life, there is remarkable diversity, but this is a famous painting by Charlie Harper. I absolutely love this painting because he did something very clever. He draws a lion and giardia. This is what gives you diarrhea when you get sick. But you see the scale are equal. It's possible to think about that individual microbes have the exact same potential, capacity, and importance as some of the iconic species that we're used to. And, a human would also, although we have, I'm going to try to refrain from saying destructive or negative things, but humans have done terrible things to modify the tree of life in all kinds of unknowing and knowing ways. But most of these sets of species, if you were to ask, have something to teach us. And we're going to go on a journey to spend some time just asking questions. I know I'm definitely going to run out of time, but I want to give you a hint of what that means, of how to navigate this tree of life for yourself. And I want to paint this picture in the background or the contrast of what is the world like today. I took this picture many years ago. This is kind of a school that I went to when I was first growing up. And even today, there are 2 billion kids on this planet. 1.2 billion of them live in families that are less than $2 a day. That would roughly be the poverty line. I mean, we're talking about half the potential, the creative potential of our planet has absolutely no capacity to directly engage in the experience of science. This is what education looks like for a significant portion. This is 500 million kids, that this is the modus operandi for schools. And my own personal experience in this journey was a high school exam that I took. I remember walking to a high school exam where I was told to draw a microscope, and you will see microscopes will show up. The irony is neither me or my teacher had ever seen a microscope. But the exam said draw a microscope, and of course I had seen it in a book. And we would draw the microscope and label all the parts. But you could not draw it from another perspective. It had to be the perspective that was in the book. So if you read the wrong book, you are in trouble. And the irony of this is that this is really what we still call life science education. And I think I'll bring this up in the very end, that this is the background. The most unknown on this planet is living, in my mind. And at that same time, we have an incredible talent and communities, young or old, having no access to actually experience it. So let's start with the number line. I'll give you example after example of phenomena that we have discovered with quite literally the first observations of them were literally made with a $2 microscope, except one or two examples here and there. And walk you through why this is actually a good framework of thinking about recreational biology. So, you know, I was going to ask you to choose your favorite cell, but then it would take me too long to give this talk. So I'm just going to choose. So first of all, geometry is everywhere. These are unicellular protists. Everybody talks about form and function. For most of the organisms, we have no idea why they're shaped this way. In fact, for single cells, we know very little about what shapes them this way. Where is the development program for shaping these sets of cells? And I'm going to choose one cell for an example. This gets quite extreme as we go to the ocean, where these are not cartoons, these are living organisms. Some of these coccolithophores are producing half of the oxygen with diatoms that we breathe, but we have no idea why they would decide to decorate themselves by these whimsical medieval weapons or something. This is calcite, so these are single crystalline structures. And I'm just going to show you this in the context of geometry for why we know very little. So let's start on a journey. All you need is a pencil, paper, a little collection jar. One of my favorite field biologists, Dan Jensen, when I went to see him in Costa Rica, said, never show up at my door without a knife, a flashlight, and a Ziploc bag. So you choose your tools of never show up in Mark's office without a pencil and a paper. Ziploc bag is an important criteria, and some of you saw what we did outside. And then, of course, I've added a little tool, which we will talk about to that. So let's go searching for some beasts. Beasts as we know them, this is what our modus operandi would look like, and much of what you're seeing was actually first observed in field, and we bring some of these systems to the lab to study. We can't grow most of them in the lab. Kind of from a mathematical perspective, I tried to give this talk a theme. You will notice by the end of the talk I'm a scatterbrain, so you don't have to be convinced on this theme, but we're going to talk about topology quite a lot because I feel it's a good way of connecting some of the mathematical ideas that are presented here. Topology is remarkable, you know, just, I mean, incredible in the context of thinking about the continuous perturbation. And I'll give you for each an example of topological invariance in a biological system, knots in biological system, orientable surfaces, and patterning of topological objects by individual examples. So here is an object. This cell is called Lacrimaria oler. It's called a tear of a swan, and you'll see for a second. I have a tradition of taking most new students in the lab and postdocs to a swamp. This is our kind of incarnation to the lab. This happens to be a swamp behind Facebook office. And we pull out our foldscopes. And there was this cell, I was looking for it for a long while. And out on one of the journeys with Scott Coyle, who was a postdoc, and Deepak, who was a graduate student, and Ellie, another graduate student, we saw this. I'm just showing you the actual footage of what we saw, although the music, oh, by the way, sometimes when we go out and it rains, this is William in the lab trying to do recreational biology in the back of a car. This is what we saw. The only thing that I've added is that the music is not real, as you'll notice. Remember, I said this is a single cell. And look what it does. You know, it's like a little arm or a neck or whatever, and it's going to search around. It's looking for food. And very soon, I know it's a video, so it's going to happen. Although sometimes we watch these things and it'll take hours and nothing happens. But, you know, you could start questioning yourself, wait a second, where is all this encoded? This looks like real-time behavior. And then, I don't know if you all notice, that poor guy just got eaten up alive. One single cell transforming itself. Now, this will play on and on. Let me show you a high-resolution version of that same video. Now, that was taken with a full scope. Now we're back in the lab, and you see this is what happens. That's a strike. This thing is a predator. And one of the important things is that it's actually neutralized the prey, and it's morphing its geometry to be able to, in real time, engulf the thing. So I know you're all scared of T-Rex or something. If you were tiny, this would be the T-Rex of the family. But it has a spectacular idea hidden inside, which is actually topological. So we started asking ourselves a question. It's a cell, so it doesn't have a brain, because a brain required neurons. But it doesn't make sense because it's a single cell. So where is behavior encoded? Where do I say, what is choosing to go where you want to go? And this is all happening real time. So we first mapped its behavior, and it actually turns out there's a beautiful scaling law. It has no blind spots, and most of the behavior can actually be explained with simple bending modes. So we knew bending of this little beam that goes in and out is important. But you know, still, that doesn't explain completely how the behavior is encoded in real time. And then we had an aha moment of asking a very simple question. What if we go back to the basics? You take an elastic beam and add activity to its tip. Now, if some of you noticed the prior video, there was cilia whipping around, so it's actually active. It's generating forces at all times. And this is Deepak in the lab, and we implemented a very simple simulation, the simplest possible thing you can imagine. The red dot is activity. It's a force that turns on and off on an elastic beam. And you run it in a non-dimensional framework, and something really strange comes out of it. Just an elastic beam starts to encode essentially chaos. So it's a very simple example of a chaotic oscillator. And you're seeing we are just plotting where it's been over time, and you see, if you can forget where you were before, you can actually encode randomness. And what it's doing, just like a good graduate student that goes searching for pizza, you know, you should only search for long enough in a place, go to another random place, didn't find food, go to another random place until you hit pizza. That's really, and then we took this model, mapped the kind of behavior, and we could actually show very subtle parameters of the blind spots associated with this behavior, only encoded in this very simple one-dimensional model. Now, just for historic reasons, and especially since there was a beautiful rocket launch recently, many of these ideas of chaos in these elastic structures were actually first discovered for rockets. There's a very simple idea hidden behind, which is geometrical, that when you apply a force on a single filament, it's a non-conservative force, the direction of the force changing. So if any of you are inclined to do this experiment, you can go tonight, open up your water hose in your garden fully. If you get wet, don't blame me, and just dangle it, and you'll see it'll actually turn into a chaotic oscillator. A very simple encoding of this remarkable behavior that you saw. Now, the puzzle doesn't stop there. When we kept looking at this, came another surprise. And this one, you know, if you believe in physical reality of the world, is a little bit hard to first understand. What you're watching is again another real-time video from wild-type cells. It's the same cell, but notice the neck. That's 40 micron, and that's almost a millimeter. But you know, where does all this material come from? A cell cannot synthesize membranes, cytoskeleton, and it's gone. It suddenly disappeared. So how many of you have seen the movie Incredibles? You know the mom in Incredibles? It's like if I was standing here and I could stretch my arm, go outside, get a cup of coffee, and come right back while also giving a talk. I would love this superpower. But this, again, was a paradox that you cannot synthesize materials, so where did that all go? Now, Ellie in the lab, was a graduate student, implemented a brilliant experiment. We decided to torture the cell. So if some of you are squeamish, you can look away. But we're going to hold the cell by its neck, and we're going to stretch it, and stretch it, and stretch it. And I'm going to stop this video because this keeps going on, and we can keep unfolding it. And in that word is a hidden answer. I said the word unfolding. And we started thinking about how could you organize membranes and cytoskeleton, which is the bones of a cell, in this manner. And you know, it's totally fine. You don't have to worry about it, because when we release it, it kind of swims away. But it has this remarkable capacity. In the entire tree of life, we have never seen a cell that can do this large of a morphological transition. So when you map the stress and strain on these sets of systems, it's the largest shape change ever recorded in a single cell. What's hidden behind? Some more high-power microscopes. The first thing we can notice is that actually the cytoskeleton structure has this incredible helical architecture. So now right under the membrane, that little tube that's sticking out, are these little coils. And this kind of looks like the Chinese basket, you know, that finger. It is not. This is a Z projection, so I've made the image that was on top and the bottom in a flat. It actually is a helix. And we only find one single chirality in nature. We have not as yet found a different chirality associated with it. But again, you know, it doesn't explain completely. We know in its folded state, all this material somehow disappears. We ended up doing another higher resolution. Now this is TEM. We're looking at nanometer scale structures. And then you end up finding these remarkable folds and these little fibers all planning around these sets of membranes. And I was on a trip to Japan, and it kind of dawned at that time, you know, of course, what we had discovered is the first example of cellular origami. And I'm going to explain this in a second. I don't know how many of you know what this object is. This is the Chochin lanterns from the ancient Chinese. They are these bamboo paper structures that fold and unfold. And, you know, I was talking to an artist friend of mine, and I ended up visiting him for some time, spent some time learning how to do this. But in the end, we actually ended up discovering that the mechanism that this cell is using is actually this folding, unfolding. It's a new kind of origami. We named it lacrigami. And what you're watching is this really strange behavior of an object that when you pull with a spring has a non-affine extension. You see, it only extends as if it unfolds with purpose. You see, it's not just randomly unfolding. And it turns out in the world of origami, these are decones. These are singularities that are actually traveling along these helix that are controlling a repeatable deployment of this origami structure. So I know some of you watched the James Webb Telescope unfold. How many of you watched that on the edge of your seat? How many of you were worried it was going to get stuck? I was worried. But that's because origami has this problem that when you deploy even one fold as a mistake and the whole thing jams. It's a classic problem that's been known. This cell folds and unfolds an origami structure 10,000 times a day, and I've never seen an error. And that's because it's actually utilizing these topological singularities that travel. So this helical folded pleated structure can only fold and unfold in one manner as these topological singularities crawl up and down of a helix. Now, some of you might have seen straws. It's important to compare this to straws. Straws do this too. But you notice the stochastic popping. Those pleats or those topological singularities are not linked, so any one of them can pop. But that is not the same structure as the one that nature discovered in this. And then there is another puzzle to it, which is the cytoskeleton, which is these microtubule ribbons that are inside, have a second twist singularity. Because remember a pleat, for a pleat to open, the normal vector of this will go from the inside to the outside. That's 180 twist of the vector, and that's happening. So you don't see it, but this twist is propagating along these molecules. So just a remarkable architecture that geometrically encodes this control. Now, we can actually watch this, we can model that, we can do all kinds of fun physics, we can measure forces on this. We are trying to do the same experiment of force spectroscopy on a cell, but the cell is not very, what's the best word? It's not a good experimental partner. This is a very difficult experiment to do because you have to catch the neck that's muddling around. And I think one thing I do want to say is one time in the history of the last 6 years I've been trying to catch the cell, I have seen one pleat defect. This is that pleat defect, and the D cones opened here, and now it doesn't know how to close it. So this will remain in this morphology for the end of the cell dying. So this was just one example of geometry, and as you will see in my talk, I will go exponentially faster in my talk. This is just my habit and looking at the clock, but I do want to share a couple other stories. You can find this cell, by the way, in your backyard or in the puddles around you. And this is the irony, that you find these incredible invention of ideas in pond scum. So this is a Santa Barbara story, so I really wanted to mention this. What you're watching, I think, are, in my mind, the cutest animals in the world. This is polarization microscopy, and you are looking at a living object that is producing single crystals. And if some of you love jewelry, which I do, they are decorated with jewels. So we'll talk about these jewels in a second. But the Santa Barbara part of it, I was at a meeting in 2017. I was kind of getting bored, and I took a walk to that beach. It was not KITP, I promise you. It was a Pew meeting a little bit further down the road, and out in my bucket, and I pulled out this little worm-like creature. And I mean, it was kind of fun. I have this tendency to look at everything under a full scope, and I found this object and I documented that. But this object that I was looking at was a mineral structure that was nothing like I had seen before. And I'm going to show you another photograph of that. So this is the object that I'd pulled out of the water. That's what it looks like. So does anybody get remembered about anchors? You know, we thought we invented anchors. I'm sorry. Ironically, these sets of structures are on the skin of a sea cucumber. How many of you have eaten a sea cucumber? You should try it at least once. And you can change your mind. They are little mineral stones on the surface of the skin. And I mean, to me, they look like weapons. And, you know, then began the journey of 6 years of work. Ironically, in biology, mineralization is everywhere. I mean, of course, I'm standing here because I'm mineralized inside, but I'm not made out of single crystals, but single crystalline architectures. And some of you who work on hard condensed matter, these objects that you are looking at, these are sponge spicules, every one of them was programmed by a living object, a living cell, and all these architectures are encoded using cell biology and cellular frameworks. And they all, although have beautiful morphologies, have a single crystalline axis. How the hell would you do that? So how do you take this and convert into that? Any arbitrary geometry. So we started thinking about it, and again, I know some of you might be thinking about snowflakes. That's not how they are built, and I'll show you a little bit of a hint. There you go. This is calcite, something that you've all played with. This is a starfish. And sea cucumbers and starfish fall into the same category called echinoderms, and this is why you can pick a starfish and it's hard, because all of these minerals then fuse together to form a giant lattice. But the thing that I want to show you is going back to the sea cucumber, and this is Pranav in the lab with Charlotte, another graduate student. For the last 6 years, we took that observation and we started asking a question: how on earth can a living object create single crystalline architectures? So you can watch, it's decorated. The beauty of it is when the sea cucumber is juvenile, it's transparent. So I can do microscopy and just watch how it grows. And this is what we saw. So these are the decorated sets of structures, and I'm just going to jump. This is the CT data to show you that there is incredible topology. It actually turns out if a cell is making something out of a rock, if you make a mistake, you cannot go back. Unlike developmental biology, when you can correct mistakes, this is patterning in rocks. So there is a one way that you can go, you can't turn back. There's all kinds of crystalline lattices, there are broken symmetries, and the thing that I want to show you, so if the seed, which is the seed crystal, has the wrong symmetry, it will actually make the wrong architecture. So these objects, although they look similar in shape, are actually topologically distinct. It's almost as if a program is running on the seed crystal, and if you start with the wrong seed crystal, you're going to get the wrong output. These crystals are single crystalline axes, and this is one of the most beautiful things I've seen in life, where you watch no matter where you look, the calcite structure has the same orientation. How the hell would biology actually control something like that? So this is now just going into the context of the best thing to always do is just observe. And this is live footage. We figured out how to mount a sea cucumber for 1 week and observe a single cell inside it for 1 week. So this is, some of you joke about watching paint dry. I'm watching rocks grow. This is 1 micron per hour. And we discovered that just like our lungs are patterned, just like our neurons are patterned, this is actually a branching morphogenesis process. And I don't have the time to talk about kind of the details of this, but in the end, the architecture is this. How would a cell know how to build a sculpture? So if you ask Da Vinci, how did he know what he was making? He would say, I would see it. And then you have feedback loop. But a cell, how can you do that? What the cell discovered is to make a sculpture, the cell becomes that sculpture. And the trick is that it wraps around this rock as a saran wrap. So what you're watching here is hidden inside, this is the rock, and the surface of this topologically complex object is the cell. And then it lays out these microtubule highways. These are roads, and these are little Amazon trucks. These are little lipid vesicles with calcium. And then you have a graph network with a growth on a graph network with a two-way coupling. So if you were to think about this, you have a graph. The road network decides where these little Amazon trucks will stop, but where you drop those trucks is going to be the consequential next graph network, and that gives rise to this growth networks on sets of patterning. So it's a really remarkable idea of growing graph networks, and then any error in that earlier version of the graph network completely changes the topology of the network. Okay, so let's keep going. I know some of you just outside saw a springtail. How many of you saw that fast insect? So speed is very important in biology. Some of you have experienced it yourself. There are times we just have to get away, and that requires speed. So not just large animals are fast. Cells, single cells, are actually much faster. I'm about to show you a video that a single cell is going to experience 20 G forces. This is a giant ciliate called spirostomum. Another cell that I was poking around in Lake Tahoe, this stumbled upon in front of my view, and I'll play this. Watch the cell. That happened in 5 milliseconds. A single cell contracting, these accelerations itself are 0.5 meters per second with 20 G forces. So some of you, I don't know how many of you tried to get into Air Force. I did. And they have a test that humans humans get knocked out with a couple of G. This is experiencing 20 G forces as a single cell. It doesn't explode. The question, and again, we started poking at it, first found it in Lake Tahoe, brought it to the lab. 2 of the postdocs in the lab that were working on completely different projects got excited about this project, and we ended up discovering a really strange thing, that these cells can talk using pressure pulses. What you're going to watch is one cell. Watch the clock here. It's milliseconds. This is high-speed video. It's sending a pressure pulse. When it contracts, the neighboring cell detects it, and it's a percolation network of communication. 0.5 meters per second, this wave is traveling across this entire ensemble, and they are literally talking using this clicking, which is a pressure pulse in the water column. They use this as a howler monkey to detect when you have a predator in the water column. And one of the things that we got puzzled after we first discovered, this is the first example of hydrodynamic communication ever reported directly in single cells. Since some of you love astrophysics, I had to bring this. We had a crazy idea. What if we did what you guys do with black holes and people, which I learned a technical term, spaghettification, which is if you get sucked in a black hole, you stretch. So we made a lab-based black hole, which is just, it's a sink flow, and all of the fluid is coming through. You can write down the potential flow. You can see that this cell will experience faster and faster and faster and faster and faster velocities. And we can watch, this is the event horizon of single cells. At the moment the strain rate is fast enough for it to click, and it contracts. And from this we can actually measure the ion channels and how these ion channels respond to forces. And one of the fun things about this is we got to write the word spaghettification in my paper, which is really why I wanted to do this. Now, going back, you find these things everywhere. I happen to be looking at a pond, and I just want to give you an idea of what would it have taken if you were sticking your iPhone around in a pond. Every one of the dots that you see in this thousands of meters of a pond is that one cell. There's literally trillions of those cells talking to each other in this massive mass of water. That particular video actually happens to be from Liberia, where we were out working on Ebola work. And ironically, some of our other work doesn't let us go. So we ended up finding these sets of cells all the way out there. Now, I had mentioned something about topology here, and I had mentioned fast animals. So this is a good recap of fast animal, really fast animal. But what truly limits this fast animal is the fact that if the cheetah was to run any faster, it's going to fracture its bones every time it strikes. It's dissipation of energy that is limiting in many of these fast systems. And one of the fun things that we actually discovered through this process is the molecular architecture inside this cell is a topologically entangled network. So there are two networks inside this cell. This is Ray Chang's work in the lab. Now you're looking at nanometer scale structures. These little threads are the endoplasmic reticulum entangled with the vacuoles, and this system provides a new kind of a break. This is jamming occurring inside the cell that acts as a break before the classic jamming transition. Because it's topologically entangled, you essentially get jamming in systems a little bit before the volume fracture, where you would, and in this geometry you wouldn't even get jamming, because this is not an isometric pressure, this is actually only a single contraction. So this was a fun new discovery because it led to a new way of thinking about how cells dissipate energy. And again, all came from that video of that first observation that this cell was doing something so fast. Ray ended up going further, and we just asked ourselves this question: in the tree of life, who is the fastest? It's kind of fun to do this because we can go out and we literally collected almost all of these species and measured them one at a time, and out came a really fun scaling law, which has 6 orders of magnitude of a universal law of speed, but not for cheetahs and animals, but for actually single cells. And the rate limiting factor is energy dissipation. Okay, I'm going to say a few words about Klein bottles, and then let me jump to some of the endings, and because I want to talk about some of the social side of this story too. Okay, so let's turn to pathogens. Up till now, everything that I told you will not actually infect you, will not hurt you in any way or form. It's going about its business. The thing that you're about to see on the screen will actually hurt you. These are microsporidia. This organism is what killed the silk route and the entire silk revolution in Italy. In the air right now, these tiny little things are actually floating, and our immune system is powerful enough to suppress this all the time, so nothing to worry about. These spores spread everywhere, but interestingly, when you're immunocompromised, this is very dangerous. But what does this cell do? When it lands on the epithelium of our eye, on our gut, it shoots out another tube. And watch this, this is a little bit slower, but within a second, you have this tiny little thing, which is only 10 micron, shoot out a tube that is 300 or so micron. Not only that, in there, right at the tip, the nucleus of the cell will shoot out of a 10 nanometer tube all the way through and inject itself into the host cell. So just crazy mechanism. And what if I told you that this cell actually doesn't have mitochondria? It has no traditional powerhouse of how we think cells power themselves. So it's doing something incredibly fast, but it doesn't have mitochondria. The answer again lies in topology. It's actually using a very clever trick of reorientable circuits and surfaces. And this is Ray and Ari in a collaboration where we primarily looked at the topology of these structures and showed that this is actually an eversion process, that the cell is turning itself inside out, and we believe it's actually using the energy in the conformation change of these proteins to power itself. So it's much more like you're lighting a fuse, and the geometry itself stores energy. Okay, so let me switch to one or two more examples, and I promise you we'll get to the end of this. This one is a fun one because it has to do with donuts. Now, we don't have any donuts. There were no donuts outside, but we have a donut right on the screen. I said this is about recreational biology. So we should be able to just ask whimsical questions. And this is a whimsical question I asked myself 15 years ago, which actually turns out has a really profound answer, which I didn't know until we found it. So first of all, I was at Caltech and foldscoping, and I saw a donut. You can see a donut right in front of you. It's a cellular donut. And I was so excited. A cell that has a topology of a donut actually has a really difficult puzzle to solve, which is how do you divide a donut? If you divide a donut, it doesn't divide the donut into two cells. So for a cell to do this, this is pretty hard. Somebody, long time ago, Ray Rappard, an incredible biologist who worked all by himself in a little hut in a coastal area in Maine, actually did this experiment. He didn't find a toroidal cell, which is what we were after, but he did jab a cell with a little capillary, forced it to be a donut, and discovered that a cell gets into a problem. When the cell divides, it divided, and you see it's still connected. But the next division, for somehow, the cell figures out that it didn't undergo two divisions and actually fixes itself. So in the next stage, it's back to normal. And this was a fundamental discovery. His playful experiment led to the discovery that spindles are detecting membranes to be able to figure out where the pinch points are. So this led to a beautiful molecular discovery, which has deep insights in thinking about how Down syndrome and many of the cells that don't complete their cell division can still have a very profound impact, and we can actually correct for it. Very playful. And, you know, I love this example of recreational biology. So to find the cell, we had to go much farther, and I'm going to now take you to the ocean, so you will get a little bit wet. All the data that you're about to see was actually collected at sea. So this is just an introductory slide for what a sea looks like when we're running and operating. We are on a tiny little ship. This moment, we happen to be off the coast of Hawaii. It's around evening 7 p.m. Adam and I, who's one of the postdocs at that time, we cast a net, and out comes something quite fun. So first of all, we spent a lot of time at sea. This is other examples of times at sea. This is in the Arctic. There is a radius you don't, so this was a little too far because you don't want to go too far because sometimes these guys show up. Not fun when you're trying to do science. They will eat you alive. But what we are after is what's actually under the ice, which is just a remarkable diversity of cells. And I'm just going to show you one of these cells to give you an example for what it feels like. So first of all, this is a soup. I bet if any of you want to find a new cell to name, go out to the ocean, collect a bucket of water, and just spend time with that bucket. I promise you, if you spend a year or so with that bucket, you have a cell that nobody has ever seen before. That's the kind of diversity that we're talking about. And I'm going to give you one example of it. So we've been doing this for a while. We searched like mad people. This is 17 expeditions in the last 5-ish years. And again, we also love spending time studying these systems at sea. Let me show you what Rahul and Adam found. We're 2 postdocs in the lab. Rahul happens to be here, so talk to him more about this. This is a paper of the discovery of a toroidal cell that's actually coming out tomorrow or day after tomorrow. This is the boat we were on. We cast a net, and we found a bioluminescent cell. Now, this cell was doing something very strange. It's a 100-micron cell, and we found it in deep waters, 200 meters, and in shallow waters. But ironically, the cell has no appendage, no flagella, no way to move. It's been called an immotile cell. So how do you as a cell move hundreds of meters in the water column and without any known appendage? So we call this, how do you move without moving? It has a surprise that has to do with topology, that has to do with the toroidal nature. So first of all, how do you even watch a cell travel 100 meters? I am from Stanford, but I love this photograph, so I'll take a hit for this for Berkeley, although we also have our own tower that's 100 meters. I have 2 versions of these slides. But you get to see the ocean. We don't have an ocean in the background. So if I want to watch a cell that is traveling 100 meters, that's a problem. I have to make a microscope that's 100 meters. I went to my chair at that time. I was like, I'm going to build a microscope at 100 meters. You can see how that conversation went. We did actually build a 10-meter microscope in Madagascar at that time, because at that time, Deepak and I were searching for parasites. So we would go out, collect some parasites, put them in these long tubes and watch how these parasites move. And then on the trip to Madagascar, we had a crazy idea. What if you take the 2 ends of the tube and connect them together like a loop? Again, donut. So we're going to use a donut to find a donut. This is the donut. We put a hamster in a wheel, but for single cells. So we built a new kind of a microscope. Now it has an infinite field of view because there are 2 points on this ring, 3 o'clock and 9 o'clock, where you can hydrodynamically levitate things. If the cell is moving down, you move the wheel up. If the cell is moving up, you move the wheel down. And its own frame of reference, it's climbing or sinking. In our lab frame of reference, it's stationary. So very much like what you all do to your hamsters. This is the instrument, and we found this cell going up and down. And that's when a big surprise. By the way, this machine, we call it for fun, gravity machine. We put not just cells in it, we put beautiful things in it. So this is sprinkles from my kids' breakfast. But as many of you study sedimentation physics, I can sync with this now sprinkle forever. So I can lock it with a computer control, and I can just sync with that object forever, watch sedimentation forever. Now, why stop at sprinkles? You can do this for your sugar. So this is the first video of a sugar as it sinks through your coffee. And you can see we can observe this because I am syncing with it, or particles that are syncing around, and you can see even non-living things do a lot of exciting stuff. This is hydrodynamic interactions. But going back to the boat, one of the things we're excited about is use that to understand. So first of all, the boats vibrate. This is another example of a cell that we put in it, and this is a very strange cell. If I was to drop this marker right here and it falls, none of you would be surprised. I'm not going to do it. It might break, but none of you would be surprised. Gravity is on all the time. Here is a cell that figures out how to turn gravity off for a little bit. And you're going to see this. The video is playing. It's a single cell. It's heavier than water. In 100 milliseconds, it's going to change its density by 5 to 10% to levitate in the water column. We had never seen a phenomena like this before. The fact that cells can actually change their density in 100 milliseconds. This is one of the fastest density change you can imagine. And we then discovered, and it does this over and over and over again. So cells in the ocean do very strange things that you can only observe when you are in the frame of reference of those cells. Going back to the toroidal nature. So this is what that toroidal cell actually looks like. It's sinking, it's rising, and one of the beautiful things that we discovered is that it actually goes, these are whale bars, so we have had to modify our figures to include whales in them, because the cell, although is only 100 micron, is traveling 200 meters. And I'm just going to show you the geometry for why this is so beautiful. The cell has discovered how to be a submarine. The cell massively inflates itself, 6 folds in volume, and that is what turns it from being sinking to rising. So it's a very strange thing to do. If any of us would inflate ourselves 6-fold, you would imagine that our density would change. It's taking in water, so it's a little bit of a desalination plant inside a single cell. And I'll just play this because this is just a beautiful video of this process at play. And you will watch, because I know this is a movie, this guy is going to inflate in a second. Now this is not in a gravity machine, a regular microscope. Boom. This is the largest volume change ever recorded for a single cell. Now for all of you, if you think hard about it, how can a cell do this? If you change your volume so much, all of the concentration, every possible enzyme you create has been diluted. Nothing should work. And there comes topology. The cell discovered a really remarkable architecture. This is the first example, as far as we know, of an N-genus torus cell. So I've now labeled the cytoplasm, and you're watching the cytoplasm is not just a bag of fluid. It's a reticulated network of cytoplasm that is linked to the vacuole. So when it expands, the volume of the cytoplasm doesn't change, although the overall cell volume changes. Without this topological trick, you could never actually do this. And I know since I'm running out of time, I'm going to spare you with some of the details of the theory for this. Actually, maybe one thing that I should mention, although I said there is nothing useful, I will say, in this talk, nothing of value, but I think this one I do want to say, because this one is important. We didn't discover it because we thought that we were going to do that. I want to just mention 2 words about climate change. We are at a very difficult time right now in the sense that we know that the kinds of changes that we have made at this point are starting to look like there is no coming back. But at that same time, we don't actually understand from the microscopic perspective of how the living parameters control most of these large-scale carbon budgets on the planet. This is well documented, lots of open physics problems. One of the ones that we focused on is actually an object that is called marine snow that was first described by William Beebe and Ortiz when they went down this bathysphere thousands of meters below, and they didn't first describe all the bioluminescent fish or anything. They described detritus, falling, sinking snowfall on our planet that happens all inside the ocean. Now that is a saving grace. And one of the important things about this is that sinking material in the ocean is how the ocean soaks up between 40 to 50% of every carbon molecule that we emit in our atmosphere. If this sedimentation was to stop, the parts per million would go from 400 parts per million to 600 parts per million instantaneously. Rahul in the lab, who's a postdoc and also back down there and has some posters on this work, we started thinking about this from the microphysics perspective. And we have just found a massive error in how we have been actually calculating and measuring this, which is worrisome. And at that same time, I'm happy we found it now and not 10 years from now. So this set of material that sinks to the bottom is really the primary mechanism the ocean sequesters. There are other mechanisms at play too, but I'm just going to focus on how objects sink. How different could it be? They should just sink through Stokes' law, right? Not really. So let's take a look. We went out to sea, and I'm just going to show you some of these videos. So in the gravity machine, we're watching an ecosystem sink. And in that process, we found something quite surprising. when we look at these objects and look at their sedimentation velocity, we didn't see a casual relationship that you would think from size. You know, often enough in Stokes' law, there is a size sinking velocity relationship. But then when we look closer, and this is the movie that I just want you to pay attention to, this is the living ecosystem of thousands of cells that have stuck and now are sinking. This is what's saving our planet currently in terms of keeping at check the carbon concentration. But look right behind there. The organisms in here are generating a parachute. So this has a mucus transparent parachute that they release, and what we ended up discovering that these mucus tails can take the sequestration, which is 200 meters per day, to almost 0 meters per day. So some places where we might think that sequestration is happening might actually completely shut down. And this is worrisome because we have no knobs, no biological knobs in any of our models. We're starting to build these sets of models, and now this paper is out, so you can read a little bit about it. But this error, when some of the friends did a rough back-of-the-envelope calculation, is as large as a fluctuation in 40 parts per million in the atmospheric CO2. So you wouldn't be happy if suddenly those sets of numbers would fluctuate by that much. Any set of technologies people are trying to build, unless we actually measure these things accurately, we would really not know. So we're flying blind in many ways because we sometimes are not measurement focused. This is the only useful thing I wanted to say in the talk. You can read the paper, much of this is online, and this is what it actually looks like. So although we all love the benthic ecosystem and literally millions of species in the ocean, all of them are supported by food which sinks to the bottom of the ocean. Okay, I think I am almost till the end. I want to say a few words on the social side of this story. As you can see, I could have told you many other things, but I promise I will end in less than 5 minutes. I have a few other students who are here, so you will hear from them throughout the meeting if you come by, but I want to come back to this. So I told you in the last 10 years of just bumbling around, you can actually find some things. Maybe useful, maybe not, but observation is what delights us when we are thinking about. How do you make that much more accessible? One of the things that we've been doing is over the years sharing these tools with the broadest group of people. I want to share 3 specific tools. We call this framework Frugal Science. And there's lots of tools that we have developed as Mark described, and maybe I'm just going to share only 2 tools. One is in the context of the ocean. This is a little microscope. How many of you own a sailboat? I know it's Santa Barbara, so I was hoping. No? Consider owning a sailboat. Consider building this open source $200 microscope, and consider contributing to measurements that we so urgently need in the ocean. Why? Because when research vessels go out, we spend between $50,000 to $100,000 a day with a lot of fuel to make measurements. No wonder we know more about the moon. You might be one of what we call citizen scientists around the world. We have 25 of them right now. I'm recruiting more. Open source hardware that allows us to measure what's happening in the ocean. As I said, measurements being important. Every one of these pictures is taken by this open source hardware. This is one of my citizen scientists. I'm going to let this play. You don't have to do this adventurous stuff. Gurek was the sailor, youngest sailor to cross the Canadian Passage in frozen ice. He travels with this instrument, collects data for us, uploads that data. He also carries a chicken with him. It's good for protein, but it also helps as company. So, you know, Moana is actually real. But this is the type of data that comes out of these sets of citizen scientists. We've shown one-to-one map between the kind of quality of data that these citizen scientists can collect versus what I or you would go and collect. This was one example. This is the type of data, all the instructions. There are hundreds of members in the community that have built these instruments, that share these instruments. And then I want to share one example, since Mark mentioned on malaria. Now this is taking a step further in health. all of these sets of open source tools that we make enable people to do serious biology. This is one instrument that we built. It's designed to look like an old Macintosh. How many of you have used an old Macintosh? I'm curious. Yes. But this object in real time from a single finger prick blood is actually detecting, so you see that's the field of view, it's moving to a field of view and it's actually detecting single parasite. The entire hardware, the entire software is completely open source. We've been starting to deploy this around 25 of these exist. We're working on the clinical validations for these. But this is, so primarily because of gaming hardware, we can actually do this in real time for the first time. Real-time detection of pathogens and parasites using microscopy. And then the last example I want to give is kind of go back to the picture and the idea that I had shared before, which is all of those were still hundreds of dollars. I often have felt that hundreds of dollars of tools still remove billions of people access. This is a tool we invented in 2011, which was the idea was to do 700 nanometer resolution at a price point of $1. That's Jim, one of my graduate students who now runs Foldscope. There is a mathematical insight here that led to the invention of this microscope. We published it as all academics. We were very happy. You know, in the world, nothing changes when you publish a paper. You wake up the next day, it's like, wait a second. Literally nothing has changed. For me, it was a little bit of a jab because something did change, which was, first of all, these are serious microscopes. So you collect, I mean, some of you saw this, the microscopy data is coming from people around the world. We had shown that these are powerful microscopes. You can watch single bacteria. But the jab that I want to show you is right here. Yes. What did change is we had posted a paper on arXiv, and a fairly well-known individual that some of you know read the arXiv paper. So beware. And he wrote $1 microscope, question mark, interesting, ... Now everything is fine. Why did he add a question mark? That kind of stuck with me. It's like, what, is Bill questioning that we have done this or it's not possible? And it was an aha moment for me personally. It's like, it doesn't matter. Unless we can give this capacity and share this to the world, nothing will change. At that point, we decided to launch a project to release 100,000 of them. At this point, there are 3 million Foldscopes. It's the world's largest amateur microscopy community in the world. People around the world sharing their own discoveries, discovering new species, finding new mathematics, finding new physics in these, all done with a community in 150 countries around the world. I'm just going to show you what some of these members look like. This is a village outside in Tamil Nadu. These are fun. This is Bay Area. You can be these parents. You see, they're not sharing the microscope with their own child. It's important to be these parents because you're just sharing what you're excited about, and then by osmosis, many people learn. There's an entire community, a global community. There are online systems that allow people to talk to each other in a kind of a peer-to-peer fashion. It spreads across every continent, and you know, age is no bar. I should have added more, but 90 plus means whatever you want it to be. I think age has nothing to do with the kind of science that you can explore. Everybody shares their lab notebook on a common platform. Many of the discoveries that I told you today that are published in fairly reputable journals, I described the original observation as a blog post literally 5 years before we even figured out. And so this is kind of a capacity to sharing stories and sharing what you observe as a lab notebook early on. And I'm going to skip most of this. There is lots of data out there. But I just want you to change your perspective of who is a scientist, who can make a discovery, who should be allowed to make a discovery, who should be allowed to see the spectacular world that we were gifted with. And I think this is a perspective that we all have to take, and especially as academics, that unless we start with observations for the community, it often enough is the case that many of them will never have a chance to engage directly in the field of science. So I want to end there maybe with a puzzle for you all. You know, this is Gurek, the chicken. This is the first animal to look under a microscope. I don't know what the chicken saw. I want to end with this one single individual. This is Mo Pandirajan. He writes in Tamil. 7 years ago, he wrote me a 2-line email when we had announced the 100,000 microscope program. He wrote in Tamil. I didn't understand it. I put it on Google Translate. I read it, and it said, I run a 12-children school. Will you send me some microscopes? I was like, sounds good. I rarely read email, as Mark knows, but I read that important email. I responded to him, and I sent him a few Foldscope. And a few days later, a few weeks later, I started talking, started thinking, and then I started shipping more microscopes. I knew he was very active. 4 years later, he sends me another email with an Excel sheet. That Excel sheet had 95,000 student names and 12,000 teacher names. And he said that singly, as an individual person, he had trained around 100,000 students. I didn't believe it. I was like, how is this possible? This is superhuman. The best way to confirm things is become Facebook friends with a person. And for the last 4-6 years, he has been running a Foldscope workshop for a group larger than this group around India every single day. That number is more than 150,000 students. This is one individual. On our community website, after English, Tamil is the most spoken language. And it sort of reminds you of the fact that he has no education degree, he has no background, he has no business in doing this, except he's deeply passionate that science belongs to everyone. Just because of him, we have one of the most thriving communities in India for this kind of work. So I want to end here. There are lots of examples of this in many countries, but Mo is very special. I'm just going to leave you with this image. I don't know if we have time for questions, but we should all ask ourselves this question: who gets to define the future of science and society? And then if any of you haven't had the chance to play with the Foldscope, come outside. I'll be here till late night. You can make your own discovery.
Yes, we do have time for questions. Robert.
That's can you say a little bit more about the marine snow and climate change? Is there any possibility of tweaking that to increase carbon absorption?
Yeah, I think the first place to start is basic science. These are phenomena, and especially the microbial knobs of our climate, we are just discovering. So whenever we're faced with a challenge, the first step should be fundamental science. And so the first thing that we are trying to do is massively increase that measurement. On a positive note, I'll say I just spent 35 days with Rahul and many other students in the lab in the California current. And I think we ended up actually discovering a cell that potentially has the capacity to increase sequestration. These are evolutionary processes. These microbes don't work for us. Some of them want to keep the carbon in the top. This particular cell loves to go deep because it can go 1,000 meters deep where nobody else can. It eats all the food and then has a way of coming up. So, I'm not saying that that's a silver lining. All I'm saying is we know very little about how our planet works when it comes down to biological microbial parameters. Another way of saying this is when you make measurements like this, you have to be very precise. You have to account for the microphysics at play, because you're going to take that measurement and multiply that by a very, 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 very large number, which is the volume of the ocean. So this process, the microbial processes are occurring volumetrically. The ocean by itself is 70% of the surface. It's the largest biosphere on our planet. So going back to this, I am a firm believer that if we care about our climate, as we start scoping and thinking about what comes next, we should massively invest in infrastructure for measurement. The United States had 30 vessels 30 years ago. These are the UNOLS Navy vessels that allow researchers like me to go out to sea and make these measurements. Guess how many numbers do we have today? 13. We are shrinking our capacity to make the most important measurement, which makes no sense. And again, I was very serious. One of you might come out of this talk and say, I'm going to buy a sailboat. I'm going to go out there, make measurements. It's very important for us to also think from the grounds up, not just from the top, bottom, what can be done, but what can be done as individuals. Any one of you could start a time series, a microbial time series right outside here in the Santa Barbara waters and actually make a contribution for that data set. That's how sort of small things lead to big things.
Yes.
The microscope that you were talking about, the DIY microscope, you just briefly mentioned, it was associated with the ocean plankton.
Yes, planktoscope.
Can you give me some keywords to search for it with?
Just planktoscope.org.
Okay.
You will find every screw, every little detail, how to build it, video instructions, a Slack community, a Discord, way too many communities than you ever want to be part of. We spend more time making this website describing this than we spend time writing the paper. So, and, you know, but outcomes of that is we just published one paper, and today, after, I would say, maybe a year and a half or two, there is around 150 of them spread across the world. And again, you know, as you notice, many of these tools still have barriers because it does cost $300 to $500 to actually build. Yes?
You've shown us some interesting little animals.
Yes.
Are you able to grow your own once you find them in the wild?
We often try very hard. With resilience and years of patience, we sometimes succeed. So for many of the things, the juvenile sea cucumber that you saw, Pranav in the lab spent almost 4 years trying to get it to be in the juvenile stage. It took a long time and resilience. The organism that you saw that we discovered, origami, we actually lost the culture recently. And so we are back on our rubber boots searching for it again, and we were the only culture in the United States to begin with. Many of these organisms either have never been cultured. That's actually true for 99% of all microbial organisms. But one of the things that ends up happening is most of it is because of lack of trying. And most of it is because we don't understand ecology well enough. We're starting to now build some automated tools for something like this. But some organisms, we do primary work in the ocean. So we will go out for 30 days, we'll take all our microscopes, our pencils and paper, and we will do the entire science at sea. Because the moment you take them out and you don't have the fresh ocean water, they're dead. We keep some of them under pressure and in darkness, so that's also useful because some of the organisms we study are found hundreds or thousands of meters deep. So I think you have to recreate the environment, which is hard.
One more question there.
And I'm around, so if anybody wants to talk, I'm here. Yes?
I wanted to ask, so you made a big point of curiosity-driven science, and I think back of how I was taught about the scientific method and how our kids were taught about the scientific method that has always seemed very prescriptive and perhaps in conflict with curiosity-driven science. Would you propose any modifications to the scientific method that we teach based on your experience?
I don't know. Let me be honest. Every practicing scientist uses this process. They poke around and they play with stuff and nothing works, and they play with stuff and nothing works, and then once in a while something works. But the reason they're poking and they have that grit is because they question what they see. So this is the scientific method. We can add all kinds of bells and whistles around it. The irony is, when we teach, we throw books at people. We throw everything that everybody else has discovered before. You have to give people the joy of discovery, and they will have the kind of resilience to sit in every lecture that you possibly can imagine. But just like you did, or some of you did. But without that, and I know all of the things that I shared today might have resonated, but they were just videos for you. The joy that we feel as scientists when you watch something for the first time is very deep. And that experience is universal. It has nothing to do with being a professional scientist. The joy of discovery is observing something. We are all part of nature. And ironically, I mean, a lot of things that we do, for example, with a foldscope, there is, other than how to use it, there is no experiment in this object. The analogy is that this is a pencil. As a pencil to a mathematician, this is to a biologist. There is no experiment, it's a piece of paper. You learn how to doodle, you can doodle with a pen, you can write equations with a pencil, you can make beautiful art, you can write poetry, but nobody else gets to dictate that. And ironically, in science, when it comes to scientific tools, we kind of create these sets of barriers thinking that people need to know what they need to do. So for us, it's very important. And again, you know, this is not just us. We've been growing this community for a while, and I've only written one paper with a foldscope. There are 700 papers in the literature, all the way from education to global health to discoveries of new species, all written by the community, of people trying their own sets of ideas. And again, you know, that was important to us. We're not trying to impose a method, just show that there is value in observation. And I think we have stripped away the capacity to observe, and we have stopped teaching how to observe. And, you know, I would say that's important in education, no matter where you are in your career or life stage.
Thank you, Manu. It's wonderful.